My name is Andy Tsai. I'm a postdoc in Dr. Tony Weisker's lab. Today I'm going to share some of my experience and my previous work based on Alzheimer's disease findings. So the topic I'm going to talk about today is the mystery of an enzyme called phospholipase C gamma 2. The variant is P522R. And then this is a mutation that associated with reduced Alzheimer's disease risk and also associated with longevity. So the topic today is that how to keep the brain young and away from pathology, because this mutation will be a very powerful tool for us to understand how brain aging linked to Alzheimer's disease. And then we know the problem for current world is that we have almost like 46 million people suffer from dementia, and the numbers keep increasing. And then around 2015, there's an expectation that we'll have 131.5 million people that will living with dementia. So we think nowadays, right, like people are getting older and older, and then we all have the expectation to live longer than 80 years old. So if you look at each other, one of you might be suffer by dementia one day, and then if you're not, if you're lucky enough, maybe you will be the caregiver. So your partners or your friends or family member maybe also suffer from dementia. So I think this is a very important issue right now that we need to find out a way to reduce the risk for brain aging and then prevent brain aging become dementia. So like at least, $159 billion has been put nowadays for treating Alzheimer's disease just for the clinic. And we haven't counted the number and the money that we put for research. So we know that Alzheimer's disease right now is a top 6 cause for death, but also people suffer from dementia or Alzheimer's disease. The average of survival after diagnosis is 4 to 8 years. So think about this. If you have a family member or friends suffer from dementia, you'll need to see their cognitive decline for at least 8 years. And then I think everyone will have a big impact on this kind of very sad disease. And then the problem for dementia or Alzheimer's disease right now is that we don't have a human model that we can help and tackle this disease. And then also the drug discovery or drug development issue is that we have very poor early go and non-go decision. For example, like some drug work for mouse model, but we don't know whether it's going to work for mouse. So the big pharma put a lot of effort on this drug. But eventually, this drug is only work for mouse, but not working for the human. And the other problem is that people have diverse genetic background. So everyone has their own mutations or environment factors, and we don't have the personalized medicine method that we can help to find out a way to slow down brain aging and dementia, because we know the highest risk for Alzheimer's disease is aging, and then also sex, and also ApoE phenotype as well. So we have environment, we have gender effect, also we have your lifestyle, and also a lot of stuff can affect this, and we don't have a right answer for this. So nowadays, we are very lucky that we have several people like donors, they donate their brain after they pass away. So we can use their brain and also use their genetic background to try to find out whether there are some mutations or whether there are some gene expressions that can link to the disease. So that's how the field are doing, try to find the mutations that will increase the risk or decrease the risk for Alzheimer's disease. And that's how the way that we try to find a target for the drug. Because if we know there's a mutation, increases gene expression, protein expression, and we can try to find a drug to target this protein and then try to see whether we can do something for preventing Alzheimer's disease or dementia. So also, we are also very lucky that the Northern group has a 100+ study going on. So they collect the people, not from the disease state, but they collect the brain from the people who are centenarians. So those are the people who live very long time and without any pathology. So we also try to learn from those people and then try to see whether they have some factors that can try to make their brain, try to make their body like young but like healthy, even though they can live like very long. So Alzheimer's disease, we also, as I mentioned before, is a very complicated disease. So the majority of Alzheimer's disease is late onset Alzheimer's disease. You can see larger than 95% are late onset Alzheimer's disease. And most of them are sporadic. And they have like genetic risk factors such as APOE4. If you have one allele for APOE4, you increase like 3 times of the risk. If you have 2 alleles of APOE4, you increase like 12 times of risk for Alzheimer's disease. And then there are so many other factors in microglia, which is the immune cells in the brain, also affect the risk for Alzheimer's disease. And the minority for Alzheimer's disease is early onset. Those are the people who are familial Alzheimer's disease, and they have like rare mutations in APP, PSEN1, and PSEN2. And the mechanism is a little bit different. The early onset model for human is that with this mutation, they will increase the A beta. And then the late onset is that you have deficiency or dysfunction of microglia, or your brain cells are not working pretty well, and they cannot really clean out the A beta production in the brain. So triggered by amyloid plaque, microglia will become activated, and then those microglia will trigger tau tangles and tau phosphorylations, and then will cause neuron death, and also you have the cognitive decline show up. And you can clearly see this beautiful image is that the blue signal is amyloid plaque, and the green signal is microglia. So you can see how microglia interact with the plaque. So based on this image, you can clearly see that microglia try to clean out the plaque in the brain, and that's their job, to try to help people clean this and then reduce the risk for Alzheimer's disease. So here I try to explain a little bit about microglia. So microglia is one of the top risks for Alzheimer's disease right now based on our transcriptional analysis. So the function of microglia here, you can see activation. Activation here, I mean, is a hallmark for brain damage to the disease, or how they work with different brain conditions. That's the word I use in the field that we use microglia activation. So activation can mean a lot of stuff, such as when you were in very young stage, early stage, you'll have synaptic pruning, and those microglia try to build your neuronal subtype, and then they also help with neuronal plasticity, help the neuron functions, and also they reprogram the cell death if there's some cell damage, and then try to clean it out. And also microglia will also phagocytosis, as I mentioned before, like amyloid plaque or other like phospho-tau, and also some other debris as well. But they also have some different functions, such as inflammation. And inflammation can be good and can be bad. So one of the ideas we have in the very beginning is how to push this microglia into a more beneficial state, and then reduce their detrimental functions. Because people might tell you that microglia activation is one of the best things that will trigger your risk for Alzheimer's disease. But actually here we try to show that microglia can also have some beneficial function. It really depends on their state and then what they are doing in the brain. So the challenge in the field is also like when you have microglia activation and Alzheimer's disease, what will be the best timing to try to manipulate this microglia function, and then when will be the right timing to give the drug to prevent the disease. And I think this is still under debate in the field. Like no one really knows what will be the right timing to give a drug, and then what will be the timing to give the therapies as well. So nowadays, last year, we have FDA approved drug for Alzheimer's disease, anti-amyloid antibody, but people are still debating when will be the right timing, and then what's the dose, and then what would be the best strategy for the patients. So I think this is what we are still doing, and we are trying to figure out what we can do to the field. So we are pretty lucky that based on the genomic-wise association data analysis, we find out there's an enzyme called phospholipase C gamma 2. This is an enzyme under the downstream of TREM2. TREM2 is a microglia receptor, which is very hot in the field that we know. Without the TREM2 receptor, microglia lose their capacity or opportunity to grab the plaque and then clean out the debris in the brain. So we are lucky to find out there's the downstream mutation in PLCG2 that associate with the risk for Alzheimer's disease. So back in 2017, there's a first paper showed that the P522R mutation, which is a hypermorphic, slightly increased enzyme activity mutation that will reduce the risk for Alzheimer's disease. And as I mentioned, PLCG2 is a downstream of TREM2, and then the function of PLCG2 is a phospholipase. So they will phospholize a phospholipid, become DAG and IP3, and also trigger the calcium pathway, and then will activate the immune response. So this is a genetic study that I told you before that the mutation will associate with reduced Alzheimer's disease risk. The odds ratio is 0.68. And then this is a functional hypermorph mutation. So if you have this mutation, your PLCG2 enzyme activity will increase. And then this mutation, if you have this, you will, even though you have plaque in the brain, it's also associated with a slower cognitive decline rate. So if you have this mutation, probably even though you have plaque in the brain, you will never get Alzheimer's disease. So we think PLCG2 might play a role in AD pathophysiology. And then we hypothesize that PLCG2 expression and PLCG2 mutation will also increase and also interact with the microglia interaction between the cells and the plaque. So the goal of the study is to understand the impact of genetic variants of PLCG2 on microglia biologies and disease pathogenesis in Alzheimer's disease. But the eventually goal is to see how brain aging linked to Alzheimer's disease and how this mutation help to slow down brain aging and the risk for AD. So we know that there's a protective mutation in PLCG2, and we are also very lucky that we find out another risk mutation in the same enzyme as well. So we have a opposite direction that we can check how the mutation affect the enzyme activity and the enzyme function and how they related to the risk for Alzheimer's disease and brain aging as well. And I think this is very thankful that we use the AMPAD consortium that I told you before. This is a huge data set, so we use a huge data set and we found this risk mutation. So then we have a chance to compare the result between risk and protective mutation in PLCG2 and then how they affect Alzheimer's disease risk. So as I mentioned that a slightly increased enzyme activity P522R will reduce the risk for Alzheimer's disease. And what is the function of risk mutation M28L? We found out that M28L is a drug-resistant form of chronic leukemia patient. So this mutation is a loss of function. So if you have this mutation, people in leukemia, they will get the treatment for BTK inhibitor, which is the upstream of PLCG2. So if you have this mutation and you got the inhibitor, it's not working because this pathway has been already shut down. So this is a loss of function mutation that people get BTK inhibitor is not working. So we know that there's some clinical evidence showing that this risk mutation is associated with increased risk for Alzheimer's disease. So we know if we increase enzyme activity, we'll reduce AD risk. But if we reduce this enzyme activity, will increase Alzheimer's disease risk. So which means that PLCG2 function is very important for Alzheimer's disease. So we also compared the human data set as well. This is a brain transcriptomic data. We compared the M28L subject with a P522R subject. And you can clearly see that most of the gene downregulated in the M28L subject is associated with inflammatory response. I think this can also help us to make it confident that this is related to microglia function because inflammatory response is a top hit for the gene expression data analysis. So what we try to do next is that we already know the data from humans, so we try to build the mouse model to find out whether the pathology as also can mimic the effect in Alzheimer's disease patients. So what we did is that we used a 5S mouse model and cross that with M28L mutation or P522R mutation, and then we try to do some outcome measurements such as the amyloid plaque deposition, microglia phenotype characterization, and also cytokines. And also we also try to use this mouse model as a small patients. So we treat the mice as a patient, and we appreciate their contribution that we use this mice to do behavior, and then we get their brain to do electrophysiology, and then we also do exactly the same thing, like they donate their brain, and then we do transcriptomic data analysis as well. So we hypothesize that even though in the 5S-fed AD mouse, the mice carry M28L variants will have reduced immune response or the bad immune response, and then the P522R mutation mice will have a beneficial microglia state and microglia function that can help reduce the pathology. So here we try to find out whether this is similar as we found in a human brain as well. So we showed that the M28L mutation is a loss of function mutation. You can see that with this mutation, the protein expression was reduced. But nowadays, we still don't know whether it's because the mutation make the protein structure change so the antibody cannot bind to that, or because this mutation made the protein degrade faster than others. And then we also check the spleen because this is immune cells specific enzyme as well. We can also show that the M28L mouse has reduced PLCG2 in spleen as well. And here is something I want to show, and I want to quote what Madison mentioned earlier. We use ImageJ to try to quantify the data. So it's not very it's professional, but it's not very efficient as well. So you can clearly see based on the brain size, right? So we have increased amyloid pathology in the mice of M28L, and we have very striking reduced amyloid plaque in the protective mouse. So we try to match the brain size, and then you can see it's exactly the same brain region that we quantify. But it's very tough to doing this in the field. So what we did is that we try to build out a new technology or skill set. So we have this first time like T2 star MRI, we imaging the whole brain. So then you have the whole brain imaging that we can quantify the plaque burden. So you can clearly see that the black dot is a hypointense signal. So the plaque sometimes will bind together with iron and other stuff, and then it's difficult for MRI to see those. So you will see the hypointense imaging. And we used this brain, right? We section the brain and do the previous imaging, sectioning the HC. You can see the color, so it's exactly the same brain. So you can see this black dot is a plaque. So the P522R mutation mouse, you can clearly see that the plaque was reduced. And the M28L increased risk mice, you can clearly see that there's tons of plaque in the brain. So this made me like very confident that the mouse is having different plaque pathology. And what we try to do next is that we also want to know whether the mutation will make microglia function very different. So what we did is that we culture the mouse microglia. We culture wild type M28L and P522R microglia, the mouse microglia. And then we treat the microglia with, for instance, A-beta, and then we wash it out to see whether they can phagocytosis, reduce phagocytosis or increase phagocytosis when they have this mutation. So you can clearly see that the M28L mouse has reduced phagocytosis, and the P522R mice has increased phagocytosis. So this kind of match our hypothesis that the increase the enzyme activity of PLCG2 with this mutation will increase the uptake capacity. So that maybe that's the reason that the plaque was reduced in the mouse brain. So what we did next is also we try to understand how the microglia interact with a plaque. So you can clearly see that based on the imaging, the M28L mouse, their microglia, they don't interact with the plaque, but P522R microglia, they have a signal and they have a signature that they engage with the plaque and then try to clean the plaque out. So this also kind of validate our previous data that they maybe phagocytosis more. And you can clearly see here, the red is a microglia, the white is the plaque staining, and the blue is a colocalization. It's pretty similar to the previous slides that you can see. There is more blue, which means that the microglia are interact with the plaque more than others. And we also check the cytokine. We can clearly see that without the interaction with the plaque, the brain has reduced cytokine release. And with the protective mouse, because they interact with the plaque more, and also there's other stuff going on for the protective factors, so the cytokine actually increased a little bit. And then compared to M28L, the cytokine was upregulated, but the M28L mouse, they don't interact with the plaque. The cytokine release was reduced. This is kind of like opposite than what other people report, that if you have increased cytokine, maybe it's bad. But we just try to show people that it's not bad or good, it's how they interact with the plaque and how they interact with the pathology. So we really need to see the functional asset to make the conclusion. It's just not based on, oh, we have more cytokines, so it's bad. The reason we have more cytokines is because the microglia try to work, they try to interact with the plaque, and they try to clean it out. And here we also showed you behavior data as well. You can clearly see that I showed you the microglia and also the plaque in P522R are different. And even though they have plaque in the brain, their behavior didn't get fat. And this is similar to the patient data as well. The patient with this P522R mutation, some of them have a plaque, but they never become Alzheimer's disease. So you can also see electrophysiology data that there's plaque in the brain, but their LTP is very similar to the B6 control. That also show that the microglia is beneficial, trying to do their work and reduce the risk for Alzheimer's disease, and also reduce the risk for dementia. And what we did next is that we try to understand the signature of this microglia. So we do a single nuclear RNA-seq with a 5' wild-type mice, and the mice with M28L and P522R. And similar to previous finding, we showed that M28L mice has reduced activated microglia signature, and the P522R mice have increased activated microglia signature. And I think this is how microglia interact with a plaque that we show in the previous slides as well. And then that's the data that we have that's showing that the P522R mouse has more activated microglia signature, but not the M28L. And also there are some microglia signature differences between activated A and activated B microglia. Even though microglia are all activated, there's also some subtype the microglia that we can identify. The function are also slightly different. And I think this is also maybe something that the morphology can also help a little bit to try to know, like, even though they are activated, but maybe they are different type of microglia as well. So what we report over here is that we use a human data set to find out the mutation associated with the opposite risk for Alzheimer's disease. And we show the microglia function are different, like they have different phagocytosis ability, different information response, and also they have different LTP data as well. And this can also help us to understand how they interact with amyloid plaque, inflammation, and synaptic loss, and also cognitive decline. So the conclusion is that PLCG2 is induced by amyloid plaque in AD, the risk mutation associated with increased AD plaque, and also the P522R mutations associated with reduced disease severity as well. And PLCG2 study might be very important for the future development of microglia-derived therapy. So this is what we have been report in the paper of immunity just online last month. So that's what we show that microglia can have with different mutation, microglia have different function. And then this also helped us to try to understand, now we know the mutation is associated with Alzheimer's disease, and also the mutation is associated with longevity. And what we can do is that we will work on maybe PBMC and also some other cell type. Then we try to understand how this mutation affects brain aging. or how the mutation make the body like young. So that's the reason that we work with Kelly and try to understand how we can learn from those cells as well. So we know that the carrier for P522R mutation has slightly increased immune response as well in their B cells. So then we try to work on the cells of the mouse. You can see the mouse with the M28L mutation, which is a risk mutation, they have reduced B1 B cells. They have reduced B cells proportion, and the same thing in PBMC as well. But the recent publication showed that the P522R protective mutation will increase the activation of B cells. So I think this makes a lot of sense. So what we did with deep cell is that we try to use a PBMC with M28L with P522R in healthy control, MCI, and Alzheimer's disease, and try to see whether we can see some morphology differences, because we already have a single cell data analysis based on those samples. So we try to understand whether we can make some association between the morphology and the gene expression data analysis. So you can clearly see here, that's what people report as well. If we do 2 times injection for the cells to the machine, they show pretty similar same data. And here's a P522R mutation and a wild-type mutation. You can clearly see that the mutation cells make the morphology very, very, very different. But we don't know like what does that mean right now. We need to sort out the cells and do transcriptomics and combine data to understand more. And then this is the data from the AI part. And also you can see here as well, if we put the MCI data, you also see a very different morphology type between the wild type and the P522R. And then you can also see here as well, like we have healthy control, we have MCI, and we have wild type, and we have P522R. You can clearly see that even though the MCI will move the cell to the right, but with a P522R mutation, the cells will keep at the left side. So maybe that means that the P522R mutation make the cells have different morphology and different cellular state, and keep them in the healthy control status as well. And you can clearly see that that's what I mentioned before, like the healthy control will be on the left, and they will kind of like shift to the right in the MCI data and also Alzheimer's disease data as well. And the P522R mutation can keep the cells in the healthy control status. So I would like to send the invitation from Deep Cell and also send to my lab. Like this is Dr. Tony Weiskorey, and then this is our lab, and then thanks for all the support from everyone. Thanks.